a paper that was recently published, SARS-CoV-2. Okay. So the spike protein is accumulating in the brain. And so here's a paper that was recently published, SARS-CoV-2 spike protein accumulation in the skull meninges brain axis, potential implications for long-term neurological complications in post-COVID-19. All right, so it says, in this study, we utilized mouse models and human post-mortem tissues to investigate the presence and distribution of the SARS-CoV-2 spike protein in the skull meninges brain axis. Our results revealed that the accumulation of the spike protein in the skull marrow, brain meninges, and spine parenchyma, the injection of the spine protein alone caused cell death in the brain, highlighting a direct effect on brain tissue. Furthermore, we observed the presence of spike protein in the skull of deceased long after their COVID-19 infection, suggesting that the spike's persistence may contribute to long-term neurological symptoms. So we're going to go through a few of the figures of this paper that are really concerning, because what they found was it didn't matter, you didn't have to be injected with the COVID vaccine in order for spike protein to actually accumulate in your brain and to stay there for a very long time. What scientists have found since is that usually if a cell is like mutated or it's having problems or it's aberrant, then if that happens, then the body tells it to commit suicide. But that is not happening here. These cells with the spike protein become these zombie cells that don't want to die, and they just keep making more and more spike protein and then throwing out all sorts of inflammatory cytokines. And so they are not dying or undergoing autophagy like they should. They are resistant to that. And so that's why certain measures need to be taken. Okay, so here in this first mouse, this mouse was exposed to the spike protein from infection. And so what you can see here, this red is the spike protein. So just from infection, this mouse has residual spike protein in his liver, in his lungs, and in some intestinal areas. So here it says, we detected spike S1 binding in most organs, including the heart, lung, liver, kidney, intestine, thymus, spleen, and pancreas. In contrast to the wild type spike 1 and one that was from the flu virus, basically, so that's what they found with it from actually getting infected. However, when they just took some spike and injected it into the tail, and so this would be more along the lines of your body producing spike and it getting into the bloodstream, you can see in this second one where it says spike S1, it went everywhere. So once the system starts making it, the spike protein itself is going to go everywhere. Now, I don't have a slide to show this, but what they have found is that there are at least 13 different receptors that allow the spike protein to get into cells. So the spike protein isn't wandering around in your blood. As soon as it's in your blood, it's taken up by cells, and it's taken up by almost all of the cells in your body. All right, and so this was an influenza protein, just showing that it co-localizes in similar areas to the wild type protein as well. All right, then what they did is they actually took sections of the brain. And here, right here, you can't really see too much spike in the brain, but when they looked closer, they did find it. But here's one that would be more analogous to vaccination, and they did see this red staining of the spike in the brain. As comparison to influenza protein, not much. So this was clearly in a rat. All right, so what they did next is they investigated for the presence of spike protein in the skulls of 34 patients who died from non-COVID related causes. So these are people that just died. And the question is, well, if they had COVID, do they have spike in their brain? And so they died during the of non-COVID related causes during the pandemic in 2021 and 2022. It says we indeed identified the spike protein in 10 of them. 213 proteins were identified to be dysregulated in the brain cortex after spike 1 injection. with 190 being upregulated and 23 being downregulated. So that's just showing you how toxic the spike protein is. When it's in there, it's really changing gene expression, and it's really doing a lot of damage in the brain. So here is their positive control. And what you're looking for is not the white, it's this kind of purplish red color that's sort of diffuse without. And so this is a positive control, all right? And then number two, these were just different people that they were looking for. Number two was, I don't know if it was a negative control, but here, number three, and you can see the purple here. So this person has spike protein in their brain. This person right here, the little purplish, has spike protein. This is a negative control. We're not seeing any spike protein. This person is having a little bit of spike protein here. Here you can see the purple spike protein. Here you can see some of the purple spike protein, and a couple of these were controls with no spike protein. So basically what this is basically showing is that just from having COVID, people were accumulating spike protein in their brain, and it is not coming out. It was there, months to a year later. All right. And then what they also, scientists have found, and this is compliments of Robin Rose, who you can see here, this was from her presentation, that the spike protein, when it touches cells, is causing them to fuse together. So only certain cells are supposed to fuse, and they're only supposed to fuse under certain very, very specific conditions. But in reaction to the spike protein, different cells are just fusing together, and they're forming these large conglomerations in which, especially if the person was vaccinated, they're then making a lot of spike protein. And so they're making spike protein, and they just are in these huge conglomerations where they're throwing out all these inflammatory cytokines that are causing all sorts of problems, mostly huge amounts of inflammation. And those, they're called syncytia, these fused cell globs aren't dying. They're not going away. They're just staying there, producing all sorts of inflammatory cytokines. So this is really a problem. If you do get them to die by giving something that causes cell death at a high level, then they blow up and they release the spike protein all over, and it just goes right into other cells and causes them to fuse and create a new zombie cell. So you really haven't gotten ahead of yourself. So you have to have something to prevent it from going into the other cells.